Hi, this is Jack Cole, and this is my assignment for reaction of kind of the microscopic imaging that is found at the Marine Biological Laboratory with Cliff Brangwyn. All right, so let's take a look at what's happening here in this video. So first and foremost, what it looks like is happening is we see this relatively homogeneous mixture of cellular substance, likely a combination of proteins and nucleic acids. And what's looks like happening is we see the formation of a new substance that is essentially coming together from smaller molecule formation. So let's see, these two combine here, this third one comes in, and we're left with not only a new cellular substance, but also one's kind of a smaller amount of what was there previously. And this is kind of the process that we know as phase separation, because we're basically seeing out of a mixture, we're seeing the formation of new substances that are kind of coming out of this mixture. And so let's take a look now at the second video. And so what we're seeing here again is we're seeing how different molecules are kind of getting attracted to one another. And this is of major importance. Let's kind of rewind it back. Let's look at this right here especially, and also down here as well. We're seeing that there's definitely potential here for the fact that various properties of some cellular molecules, such as how proteins have their R side chains, how that is playing a major impact in what's coming together. And this is of major importance because cell functioning and a lot of what it does, things like protein formation, gene expression, cell signaling. This is all dependent on some of the the cellular components. And here what we're seeing is that some are being attracted to each other, again, in this phase separation process, leading to the formation of larger scale kind of substances within the cell. And lastly, let's take a look at this other video here. What we're seeing, and this possibly be evidence of, is the fact that most of the organelles that we know of are membrane bound. And what that means is that the organelles like the nucleus, the endoplasmic reticulum, the Golgi apparatus, etc., have membrane outer components are membrane bound. That means that they're surrounded by phospholipid bilayer membranes. But what this is showing evidence of is potential new even possible like organelle compartmentalization that's happening without the membrane component to it, because again, this is just a combination of proteins, nucleic acids, and some other substances that are not necessarily lipids and not necessarily part of the phospholipid bilayer. And so this is important for a lot of reasons, but especially because the way that, as I said, the cell functions and different cellular processes are going to be heavily impacted by the way that the cell is organizing itself, and especially some of its larger molecules. And then one other thing too is this is important for compartmentalization, which is how the cell basically regulates the various processes that it needs to undergo. And essentially, again, without the necessity of membranes, if a cell is able to regulate its processes, it's going to be a lot more efficient. And looking at kind of these specific interactions between these molecules can really help, not necessarily, talk to us about the membrane component, but the separation, the phase separation that leads to not needing necessarily that membrane. So thank you for listening, and I hope this was helpful.